Hello everyone and welcome to today's episode. In our previous episode, we discussed about identification of methanol and ethanol in alcoholic beverages. In our today's discussion, I want us to go through and learn how to use a gas chromatograph to identify and quantify methanol and ethanol in alcoholic beverages. What is the principal operation of a gas chromatograph? A gas chromatograph is an analytical instrument that measures the content of various components in a sample. The analysis performed by a gas chromatograph is called gas chromatography. The principle of gas chromatography is very simple. The sample solution is injected into the instrument and enters a gas stream which transports the sample into a separation tube known as the column. Helium or nitrogen is used as the so-called carrier gas. This is the schematic diagram of a gas chromatograph. This is where you have the carrier gas. The carrier gas is basically helium or another gas which is inert, which we call nitrogen. This is where you position the carrier gas. The carrier gas carries your sample, which is here. You eject your sample here in the injection port. Then the sample goes through this column. This is the column. This column is where the separation takes place. And the column is positioned inside an oven which has higher temperatures. Then when the sample undergoes separation in the column, the response is detected by the detector here, and this detector passes the information to your computer or PC for analysis and quantification. Basically, this is a clear picture of a gas chromatograph. This is an auto sampler. This is where the oven is here. Then the injection port is somewhere here. The detector is somewhere here. And this is your computer, which helps in quantification of the results and giving you the reports as per the machine analysis. Now, what does the detector do? The detector basically gives you a response of the concentration of your specific analytes. Analytes are the different components that makes a particular sample. For instance, in this sample, we have component A, B, C, and D. And the separation of all these takes place here in this column. And the separation takes place according to the boiling point of these various components of a particular sample. Now, a graph which is showing the detector response against the time, which we call the retention time, is what we are calling a chromatogram. And a chromatogram is basically a two-dimensional plot with the ordinate axis giving concentration. This is the ordinate axis, this one. Then we have the abscissa axis, which represent the time. This is the abscissa axis, which shows the time. Time in this is the retention time. The time taken for this particular analyte, this one, this particular component A of a sample, to get out of the column. Remember, the injection is here. Then the sample goes through the column and it elutes from this column heading to the detector. The time taken between the injection port and the detector is what we call retention time. And once an analyte has been eluted or comes out of the column, the detector detects and sends a signal, and that signal is being amplified into a peak. This is a peak. So a series of peaks based on the various chromatograms, based on the sample is what we are calling a chromatogram, a graph showing the detector response in terms of concentration of various component of a sample vis-a-vis the time taken for each and every component to elute all to come out of the column. Specifically, when we analyze methanol and ethanol in a sample, for instance in an alcoholic beverage, this is how the chromatogram of a methanol and ethanol is going to look like. You note that methanol elutes first, that is, it comes out of the column first as compared to ethanol. Remember, all this separation is taking place here in this column. So when you're analyzing an alcoholic beverage which you suspect contains methanol or ethanol or both, and you want to quantify and know how much of the ethanol or the methanol is present in your alcoholic beverage, your GC, or what we call gas chromatograph, is going to give you a chromatogram which looks like this. This one is the concentration, and this one is the time taken for methanol and ethanol to elute out of the column. You get it? And why does methanol and ethanol has different peaks? The boiling point of methanol is lower than the boiling point of ethanol. The boiling point of methanol is 64.7 degrees Celsius, while the boiling point of ethanol is 78.37 degrees Celsius. So basically, when a sample contains both of these components, if your alcoholic beverage contains both of these components, then methanol is going to come out of the column here. It's going to come out of the column, and it is going to be detected faster or earlier as compared to ethanol. So basically in a chromatogram, you'll have two peaks, one for methanol, it should be the first one with a very low retention time, and another one for ethanol with a bit higher retention time, basically because of their differences in boiling point. Now, this one is detection. Your GC or gas chromatograph, this machine, has detected your sample, your alcoholic beverage, to have methanol and ethanol. Now, the next thing is to quantify how much of your methanol and your ethanol is present in your alcoholic beverage and be able to determine whether your product is fit for human consumption or whether the ranges of concentrations of both methanol and ethanol are within the acceptance criteria as described by the Kenya Bureau of Standards. Fellow chemists, fellow manufacturers, I hope you are more informed on this. Goodbye, and let's meet next time in our next episode.